Hello all the students and viewers. In this video we will be studying about comparative account on dicotyledons and monocotyledons. Before studying comparative account on dicotyledons and monocotyledons, we need to know some of the general characters of angiosperms. So let us start with etymology. So the word angion is the first word and the sperma is the second word. Angion meaning vessel and sperma meaning seed. So vessel is the word here which came with the context of fruit. So seeds are covered with fruit, that is called as angiosperm. In simple words, we can say that angiosperms are seeds as well as fruit bearing plants. Now we'll have a look over general characters of angiosperms. So angiosperms are flowering plants. They show greatest diversity in size, shape, occurrence, habit, and lifespan. So they are ranging from few millimeters like Ulfia up to hundreds of feet like eucalyptus. Shape is also differing from species to species. Their occurrence is they are hydrophytes, they are xerophytes, they are halophytes, they are lithophytes, they are growing on another plants as parasites, they are taking just support of another plant, that is called as epiphyte. Their habit is different, they may be herbs, shrubs, trees, climbers, and their lifespan is also differing from species to species like ephemeral to perennials. The sporophytic plant body shows distinction of root, stem, and leaves. The plant body is characterized by presence of vessels in the xylem. This is very important anatomical character of angiosperms. Presence of siphonogamy. Siphon means tube and gamy means the gametes. So these gametes are actually carrying, male gametes are actually carrying towards the female gametophyte through a tube-like structure that is called as a pollen tube, and that is called as a siphonogamy. And another characteristic is there that there is a double fertilization. Then endosperm is triploid and developed after fertilization. This is also very important peculiar characteristic feature of angiosperms. They have complex and advanced reproductive structures, that is flowers. So these are few of the important characteristic features of angiosperms. This figure is giving much information about angiosperms. So if this is a ground, below the ground, here you will see the root system, and above the ground here you will see the shoot system. So here primary root is there, secondary and tertiary roots are there, leaf is there, axillary bud is there, apical bud is there, flower is there. So these are few of the parts of angiospermic plant that we need to know before studying their comparative account. So now let us study the comparative account of angiosperms. We will start with the root. If you see in monocotyledonous plants, see what kind of roots are there. There is a fibrous root system. You might have seen in jawar, you might have seen the roots of wheat. There is a fibrous root system. And if you see in dicotyledonous plants, where embryo is with two cotyledons, here embryo with single cotyledon, and roots of this dicotyledon plant, here you can see that there is a taproot system. Taproot systems which are growing very deep into the soil, and it will have, this is the primary root, these are actually secondary roots, and these secondary roots again have branches. These are called as a tertiary roots. So This is the difference in roots of dicots and monocots. Now we'll see the further details. Here you can mark the difference between two roots. If you look into the first picture where monocot root is represented, and here you can see a stellar region. I think I need to change the color. So if you look here, this is a stellar region, and if you look here, this is a patch of xylem, this is flat patch of xylem, this is patch of xylem. So if you see, this is multiple patches of xylem are there, and that is called as a polyarch condition. But on the other hand, if you look into this figure, here there is 1, 2, 3 and 4 patches of xylem are there, and that is called as the tetrarch condition. So among dicots, it may be triarch or it may be tetrarch. But apart from this, there is no possibility. But if you look here, this condition is polyarch. So this is the difference in dicot and monocot roots. Now let's have a look over difference in dicot and monocot stem. So if you look in the first picture in dicot, here you can see branches. So the stem has lots of branches. But if you look here, this stem is throughout its length, it is totally without branches. So note down the difference that stem here is branched, and in this case it is unbranched. That is the difference in stem of dicots and monocots. Then here we will see the anatomical differences in dicots and monocot stem. So this is dicot stem and here is monocot stem. If you cut this dicot stem in transverse section, you will get picture like this, and here you will see vascular bundles. Say for example, let me draw here 1, 2, 3 and 4, and these vascular bundles are arranged in ring. And similarly, if you cut monocot stem in a section, you will see these vascular bundles here like this. They are scattered anyway. There is no any particular pattern of arrangement of vascular bundles. So they are scattered throughout the stem. So this is actually the difference in dicot and monocot stem. So this is dicot and this is monocot. Now we will understand the differences between dicotyledons and monocotyledons vascular bundle. If you look in this picture, this is vascular bundle of dicot and this is vascular bundle of monocot. In this case here, you will see this tissue that is called as a xylem. You will see another tissue that is called as phloem, and in between xylem and phloem you can see this tissue. That tissue is called as cambium. Now cambium is important with respect to growth, and due to presence of cambium, this is open for growth. This is open for growth, and so that this is called as open vascular bundle. Whereas if you look into this monocot, see you will see xylem, this part. You will see phloem, this part. But if you look here neatly, here anywhere, you cannot see a patch of cambium. So due to absence of cambium, this type of vascular bundle is called as closed vascular bundle. This is called as a closed type of vascular bundle. Now, as there is absence of cambium, there is no possibility of further growth. So that in monocotyledonous plants, secondary growth is absent, and due to presence of this cambium in dicot, secondary growth is present. Here you will see the difference between dicot and monocot leaf. So first picture is here, that is showing dicot leaf, and this is of monocot leaf. If you see, here is the midrib and lateral veins are arriving from that midrib, and this is called as reticulate venation. And if you look here this way, there is parallel venation. This is actually the difference in dicot and monocot leaf. Here you will see anatomical difference between dicot and monocot leaf. So if you look into this dicot leaf, this is upper epidermis, this you can see. This is lower epidermis, you can see. And this entire tissue is called as mesophyll tissue. But if you look neatly here, these cells are columnar cells. And this columnar cell is called as palisade tissue. These columnar cells are there together forming a tissue that is called as a palisade tissue. And if you look here down, here are spongy cells, and they are forming together a tissue that is called as spongy tissue. Similarly, if you look here in monocot leaf, you will get upper epidermis, this layer. You will get lower epidermis, this layer. But as you have seen this mesophyll tissue here you can see, but as you have seen that here is the palisade tissue and spongy tissue, such type of tissue differentiation is completely absent here. Throughout the length, you can see all tissues are alike. So you can either watch leaf from this side or you can watch leaf from this side. It shows similar kind of structure throughout its length, and this type of leaf is called as isobilateral. Why this is called as isobilateral? Iso means similar, bi means two. This is one and this is two. So, and lateral. And if you look here, there is no similarity throughout the length, so that this type of leaf is called as dorsiventral. This type of leaf is called as a dorsiventral leaf. Now another anatomical difference is there, that in case of dicot, here you will see structure of stomata, and in case of monocot, see how is the structure of stomata. Here the guard cells if you see, their shape is like a kidney. So kidney shaped gall cells are there. And if you look here, they are dumbbell shaped. So this kind of shape generally seen in monocot. This is one and this is another gall cell. So they have dumbbell shape. This is another difference in both. Here we can see again few of the differences with respect to flower. If you see in monocotyledonous flowers, they are mostly trimerous, and here dicot they are either tetramerous or they are pentamerous. Tetramerous and pentamerous means the floral organs are in the multiple of either 4, or pentamerous means 5, and trimerous means in the multiple of 3. Here you will see difference in cotyledons and seed. So in monocotyledon, here you can see embryo, but that entire embryo is covered with single cotyledon. Here somewhere embryo is present and that embryo is covered with single cotyledon. Whereas in dicotyledonous plants, here you can see this is an embryo, and this embryo, when seed is closed, it is covered by two cotyledons. That is the difference in the seed of dicots and monocots. Here you will see the comparative account with respect to various parts, various organs. First of all, you will understand seed. So seed here in monocot it is with one cotyledon. In dicot it is with two cotyledons. It is roots are fibrous in case of monocots. They are tap rooted in dicots. Then vascular bundles in monocots are scattered, and vascular bundles in dicot are arranged in a ring format. Leaves of monocots with parallel venation that you can see, and leaves of dicot here you can see with a reticulate type of venation. Flowers of monocots here you can see that they are trimerous, that is in the multiple of 3. Here you can count 1, 2, 3 and 1, 2, 3. And here you can see either tetramerous or pentamerous, means either in the multiple of 4 or 5. You can count here 1, 2, 3, 4 and 5 petals can be seen, and here something is showed. You can consider that as a stamen, so they are also 5 in number. This way we have seen the differences between dicotyledonous and monocotyledonous plants with respect to root, stem, leaf, flower, seed, cotyledon, anatomical differences with respect to root, stem and leaves. Thank you.